budding biologists, and welcome to the Module 1 and 2 review part 1. We're starting off by looking at the cell theory. Recall that cell theory states that all living things are made of cells, cells are the basic units of life, and cells come from pre-existing cells. So let's explore first the two types of cells. They fall into two main categories, prokaryotic and eukaryotic. Remember, prokaryotes have DNA in plasmids and they have no membrane-bound organelles, where eukaryotes have membrane-bound organelles, things like mitochondria and chloroplasts, and they have DNA in the nucleus, in that helix structure. Eukaryotes are broken down into plant and animal cells. While I quickly draw this up, now we're going to look at the organelles that are common and different between plant and animal cells. First, let's start off with smooth and rough ER. They create proteins and package proteins to be used by the cell. Then we have the nucleus, where it's the location of DNA storage and coordinates all the cell activities. Next, we have our cytoplasm. Remember, that's where some of our chemical reactions happen. And then we've got mitochondria. Remember, mitochondria are the location of respiration, and that's how the cell produces energy. So let's look at cellular respiration in a little bit more detail. Recall that the equation for cellular respiration is glucose plus oxygen gives ATP, carbon dioxide, and H2O. It's helpful to know both the written word equation and the chemical equation. Respiration produces ATP, which is the type of energy that then can be used by the cell for other purposes. Next, we're going to look at the chloroplast. The chloroplast is found uniquely to autotrophic cells, and it uses light reactions of photosynthesis to create chemical energy. Recall that the word equation is carbon dioxide plus water in the presence of light produces glucose and oxygen. There's 4 more steps that we can review at another time. The next organelle that we're going to look at that's unique to plant cells is that of the vacuole. It is quite large in a plant cell and is responsible for water storage and waste breakdown by enzyme activity. Animals can have vacuoles, but they tend to be much smaller. The next structure unique to plant cells is the cell wall, and they are cellulose dense, and they provide the structure and protection for the cell of the plant. The last organelle that we're going to look at between the plant and animal cell is the lysosome. The lysosome is the location of chemical digestion, primarily that of enzyme reactions within the cell. Next, we're going to look at the cell membrane, common across both plant and animal cells. Quickly draw that up. We need to remember that the cell membrane is a lipid bilayer, which means that it's made of phospholipids, and it's got studded with protein channels, and it's based on that fluid mosaic model, which you can review in more detail in one of my previous clips. There are 4 main types of transport through the cell membrane. There's diffusion, where things pass straight through. There's osmosis, which is the movement of water molecules through the cell membrane. And then there's facilitated diffusion, where there's the use of a protein channel or a pump for materials to reach equilibrium. And then there's active transport, where there's energy required. Each mode of transport allows for different materials to pass through. So diffusion is for small non-polar molecules, for example oxygen, carbon dioxide, nitrogen. Whereas diffusion osmosis are small uncharged polar molecules, for example water or glycerol. Facilitated diffusion is for large polar molecules, so things like glucose and sucrose, and active transport requires energy, and that's for things like ions like sodium, potassium, chloride. Now let's look at the concentration gradient. So either side of the cell membrane there is either going to be a higher or lower concentration. So with the concentration gradient, so from high to low, that's passive transport, where from low to high, that will be active transport requiring energy. So let's look at this in a little bit more detail using this little concept map. So we've got two types of transport, passive and active transport. Passive transport is with the concentration gradient, active is against the concentration gradient. Active requires ATP. So against the concentration gradient can either be in vesicles, exocytosis exiting and endocytosis entering, or it could be the use of a pump. Let's look at a little bit more closely. Phagocytosis is an example of endocytosis, and so is pinocytosis. Pinocytosis is the entrance of liquid materials into the cell, whereas phagocytosis is the entry of solid materials. Now, passive transport. First off, diffusion. Diffusion is the movement of materials from high concentration to low concentration to reach equilibrium across the cell membrane. Next is osmosis, which is the movement of water from high concentration to low concentration to reach equilibrium. And we're going to explore water balance in different types of solutions and the impact on osmotic movement. Cells can be in three types of environmental conditions: hypertonic, isotonic, and hypertonic. Hypertonic solutions, the concentration of water is less on the inside than it is on the outside. So that means water moves into the cell. So the cell will expand as water moves into it. Isotonic is where the concentration is equal and water remains constant, moving in and out of the cell. And hypertonic, outside is higher, so water will move out of the cell and the cell will shrivel up. The last type of passive transport is facilitated diffusion, and it's the use of a channel or a pump to reach equilibrium of the material on either side of the cell membrane. Now let's look in a little bit more detail of the type of materials that are used by the cell. Carbohydrates, they're a source of energy. Glucose would be an example. Lipids form hydrocarbon chains, they store energy, and they can be used for membrane structure. DNA, that double helix, found in nucleus, is used for heredity and coding DNA, and proteins are in amino acids that can be for structure, membranes, antigens, antibodies, and enzymes. We looked in great detail at enzymes as biological catalysts that reduce the activation energy required for chemical metabolic reactions within organisms. So remember this graph of activation energy where the y-axis is the energy required to complete a reaction and the x is time, and we'll graph both with and without an enzyme. So without an enzyme, there's actually a higher requirement of activation energy to cause the reaction to occur compared to with an enzyme. The other key thing that we talked about with enzymes is that enzymes act on a chemical reaction, but they're not consumed by the reaction. And what that means is they're able to be used over and over again, which is very useful for completing metabolic reactions. So when we look at the picture that I'm drawing below, we understand that enzymes have an active site in which bonds with the substrate. The substrate, when it's bonded with the enzyme, is called the substrate enzyme complex. The reaction happens and it produces the two different types of product. Next, recall that there's a whole lot of different enzymes that carry out a range of different chemical reactions. We need to also recall the lock and key model and the induced fit model. Make sure you check out any resources that I previously gave you on those two different types of models about enzymes, and also review inhibitors. There's three main factors that we looked at in terms of affecting enzyme activity. We looked at rate of reaction and pH, where there has an optimum zone. We looked at temperature and substrate concentration. Oops, got them the wrong way around. Next, we're going to look at the hierarchical structure of organization of organisms, and it relates directly to all living things are made of cells, and all cells come from pre-existing cells. Let's start off at looking at cell differentiation. So, cell process of becoming more specialized. So, for example, stem cell becoming a blood cell. So, cell differentiation results in specialized cells that have specific functions for a multicellular organism. And so we recall that, organelles make up cells that make up organs, that make up tissues, that make up organs, that make up organ systems. And I've given you a plant example. Now, the last little section that we're going to look at relating to cells in this clip is looking at observing cells. So we compared the light microscope with two different types of electron microscope, SEM and TEM. And we're just going to complete a little table summarizing the main concepts for you of the different types of technology. I should use a ruler, shouldn't I? So first off, the type of energy source, and we're going to look at the image, the use, and the positive and negatives. So light microscope uses light and it's focused with a lens. It produces a coloured image. It's relatively low magnification. It's used for live samples or things where we're looking at things that are quite large. And the advantages are that it's cheap, you can look at live organisms, it's accessible and coloured, but it actually has relatively low magnification and resolution compared to its electron microscope counterparts. Both SEM and TEM use an electron beam focused with electron magnets, and they both produce highly detailed images. SEM produces images of a surface, whereas TEM produces internal images that are highly detailed. SEM uses a gold stain, which gives a high quality image, where TEM uses heavy metal staining to show high quality images of within the cell, so things like lead or mercury. The advantages of SEM and TEM is they both have very high magnification and resolution, but the negative is that it's a dead sample, so we can't observe any processes that are happening as if it was alive. They're both very expensive, and for SEM it's only a surface image, whereas TEM, the limitation is that it's a relatively small field of view. I hope that helped your understanding of Module 1 and 2 looking at cells, and I hope to make Part 2 soon. See you next time.